All right. So can everybody see that? I hope it's not super stretched out, is it? It's good? Okay. All right. Because sometimes the words get skewed on here. Okay. So it says chapter 10 in the current textbook is chapter 11. Regardless, what we're covering today is going to be muscular tissue, right? And this will lead us into our discussion on specific muscles. So select muscles of the body, skeletal muscles specifically. So that will be something we'll talk about tomorrow in lab. Today we're going to talk about muscular tissue. Now, before we talk about muscular tissue, I just want to jog your memory. I know we had a break in between, and it's probably been a while since we covered this concept. But does anybody remember the four different tissue types? Four different tissue types. You can put your answer in the chat box if you'd like. You can. I got some here. Remember that all your body's made up of fundamentally it's made up of atoms that are connected together to give you molecules, which give you cells. Then cells come together to give you tissues. Now there's four different tissue types. We have connective tissue, that's one, right? We have epithelial tissue, that's another. Muscular tissue, obviously, that's going to be another one, right? So a muscular tissue, and then there's the last one's a nervous tissue. Okay, so four different tissue types. You have muscular, connective, epithelial, and nervous. All right. So just a brief summary of each of those. When you're talking about epithelial tissue is made up of cells that are covering or lining a surface. Any surface of your body, it's going to be made up of epithelial tissue. It's basically a barrier that maybe lines an organ or the inner wall of an organ. Right? That's epithelial tissue. Connective tissue is the stuff that holds various tissues together and organs together, and you find connective tissue in the broadest. It's considered the broadest type of tissue because of how diverse the different types of cells are. Blood would be considered connective tissue, cartilage, bone. Plenty of different connective tissues that exist in the body. The two of utmost importance in this, at least in this course that we're going to really hone in on, can be muscular tissue and nervous tissue. So today we're going to talk about muscular tissue and the three subtypes of muscular tissue. Now, something to keep in mind with muscular tissue is made up of muscle cells. So because tissues are all made up of cells, muscle cells are going to be the cells that come together to give you muscle tissue. All right? Does that make sense, guys? Muscle cells, right? You have a bunch of different muscle cells that come together to give you muscular tissue. Now, there's three types of muscular tissue, and somebody mentioned one of them here in the chat box. One of those would be cardiac muscle tissue. The other would be smooth, and the one that we're probably most familiar with is skeletal, all right? So three types of muscular tissue. Something very important to note about these muscle cells, and I say muscle cells, but they're actually called muscle fibers. So just keep that in mind. Whenever I'm referring to a muscle fiber, I'm talking about a muscle cell, okay? Now, skeletal muscle fibers, smooth muscle fibers, and cardiac muscle fibers, there's going to be some distinctions there, but if you hear the term muscle fiber, just keep in mind that we're talking about muscle cells, okay? Now, as I was saying, there's something very important to note about muscle cells as well as the cells that make up your nervous tissue. What are the cells, if you had to guess, you may have not covered this in extensive detail, but what are the cells that make up our nervous tissue? What are some cells, or at least the key players in our nervous tissue? You can take a guess at this. It's not because we haven't covered it exhaustively. So dendrites and axons are part of these cells. Neurons. So, Jasmine, that's a great answer. Dendrites and axons, they're actually the structures of those cells. They're like basically components of those cells. But those cells that I'm interested in when we're talking about nervous tissue, they're called neurons. Okay. So why I mentioned neurons here is neurons, their function is to convey electrical signals. And these electrical signals are communication signals that tell muscles when to do what they do. So what do muscles do? They contract. They contract in response to electrical signals. That's why there's a very close relationship between neurons and muscles. And we'll talk about that today. We'll talk about what's known as the neuromuscular junction. In fact, if you're picking up your pen right now to write down some notes, there was a signal, it's called an action potential, that originated from your brain, and it traveled down the nerves to the muscles in your hand to pick up your pencil to then coordinate a writing motion, right? That's how there's a communication that's taking place throughout the body, and that's occurring by way of the nervous system. It's telling the muscles of your body to contract, right? So we'll get into that in more detail. I know we're on the opening slide. I'm saying all this information, but just kind of setting the foundation here before we get into all this detail. Everybody should be familiar with the four different tissue types. And you should definitely know the three different muscular tissue types, right? The subtypes, I guess you can call them. Okay, so just keep that in mind, and we'll keep on going with this on this topic. Okay, so three types of muscular tissue. We have skeletal muscle. This is, like I said, this is the one that most people are familiar with. The function of skeletal muscle is to move the bones of our skeleton, motility, right? And some move other skeletal muscles and some move skin. So skeletal muscles are all connected to either bone, skin, or other skeletal muscles, which is quite interesting. But what we're most familiar with and what we can associate with skeletal muscles is our movement of our skeleton. Now, this happens when your skeletal muscles and the fibers therein, they contract. So when they contract, what's another way to refer to contraction? If I'd ask you, definition of contraction, what's happening there? Something contracts. What's it doing?
Shortening.
Shortening. Perfect. Shortening. So when it shortens, right? When it shortens, it pulls. It pulls on the bones that it's attached to. Okay, that's skeletal muscle. Cardiac muscle. These muscle fibers, they're only found in the heart, right? So for most of the heart wall, we also have some connective tissue there, but cardiac muscle is going to only be found in the heart wall, or in the heart, as the name implies. Smooth muscle is another type of muscle tissue, and this is going to be found in the walls of your hollow organs. So your hollow organs would be things like your stomach, for instance, right? The wall of your stomach. Well, we know the stomach, it churns and it mixes. It has some motility associated with it. So the reason why it is able to move and churn and actually contract and relax is because of the presence of smooth muscle, all right? And we also find smooth muscle lining the walls of our blood vessels. Now, why might this be important? What's going to happen when when the smooth muscle fibers in our walls of our blood vessels, when they contract, what's going to happen to the blood vessel? Think about a blood vessel as like a tube, right? It's going to constrict, right? If something constricts, the diameter gets smaller. That means that it can impact blood flow. So it's really quite interesting when we consider, muscular tissue, it's beyond just moving your body. A lot of things are happening as a result of the action of muscle tissue. Depending on which muscle tissue we're talking about, we can look at very specific organs and get an idea of what is going on with those organs when that muscle found within those organs is contracting. All right? Does that make sense, guys? The three different muscle tissue types and where we find them? Everybody okay with that? All right, we'll keep going. Some distinctions to be made here then. Okay, so skeletal muscle. Skeletal muscle, again, it's going to be found associated with your skeleton. Its functions to move bones. Now, if you look at an individual skeletal muscle fiber, one cell, let's just say you took your bicep and you zoomed in such that you're looking at just one cell in your bicep muscle. What you'd find is that it's multinucleated and it's striated. Okay, multinucleated just means it has many nuclei, right? Striated refers to the fact that it has this banded pattern, this alternating pattern of light and dark bands, and we'll talk about why that is. These bands actually have to do with the skeletal muscle's ability to contract, right? And we'll talk about why that is in a little bit. But just keep that in mind. The appearance of skeletal muscle is that it's multinucleated, many of these nuclei, and it's striated. Okay, you guys may have seen striations, like if you go to like the supermarket and you see like a slab of meat, and you can see those lines on there, right? Those are striations, right? So, and functionally speaking, when we look at skeletal muscle and how we can control and actually get our skeletal muscle fibers to contract, well, this is under voluntary control. So again, going with your bicep, right? The example of your bicep. You can voluntarily control it when you contract it, right? You can voluntarily control when you get up and walk out of your chair, or you pick your pen up and write. All your skeletal muscle is under voluntary control. You have to think about it in order to get it to contract. Barring some instances like shivering or twitches, obviously those are involuntary reflexes, but your skeletal muscle, because we're able to voluntarily get it to contract, it's designated as being under voluntary control, okay? We can do so with volition, if that makes sense. Cardiac muscle, on the other hand, just on the note of control, well, we don't have to think about our heart contracting, right? We don't have to think about our heart pumping. It's under involuntary control. It happens without our volition, without our thinking about it. It does so on its own. Okay, so something to keep in mind there. Right, cardiac muscle is under involuntary control. Now, of course, if you start thinking about something that maybe like a scary movie or something like that, or like a um, it's something that gets your heart rate rising, right? What you're not actually initiating the cardiac muscle from to start contracting more rapidly. What you're doing is causing a change in your biochemical makeup that's resulting in an increase in heart rate. At the end of the day, your cardiac muscle is under involuntary control. All right, everybody keep that in mind. Everybody okay with that? The concept under involuntary control, your cardiac muscle. If you look at the appearance of cardiac muscle, an individual fiber or muscle cell in cardiac muscle, what we find is that they're mononucleated, just one nucleus. Also striated though, okay? So you also have this banded pattern in your cardiac muscle. And what we also find are these intercalated discs, right? These intercalated discs, you can kind of see them here. Zoom in, you can see how this one cell is connected to the next cell at this junction. Well, this junction is known as an intercalated disc rather. So this is what holds neighboring cardiac muscle cells together, are these intercalated discs, okay? They consist of what are known as desmosomes. You guys remember desmosomes from last session? We talked about gap junctions and how cells are basically bound together. So gap junctions and desmosomes are going to be hallmark features of intercalated discs. All right, there are some gap junctions here that, obviously, if you looked into this and actually zoomed in much more, you'd see that there are some channel proteins that allow for ions to go from one cell to the next. So, just harping on the importance of gap junctions is exactly that. They allow cells to communicate or to transfer nutrients between one another. And we find these here in these intercalated discs. You have these gap junctions, okay? So keep that in mind. Gap junctions are important in these intercalated discs because these neighboring cells need to send ions from one place to another and nutrients. All right? Does that make sense, guys? Why intercalated discs and what intercalated discs are and why they're important? Because this cell is not able to communicate to the next cell, and this one just contracts, but it can't communicate anything to the next one, then the next one's not going to contract. And being that this is part of your heart, well, you can't have muscle fibers in your heart not contracting, right? That's going to lead to heart failure. So very important to keep in mind the importance of those gap junctions. Okay. Now, overall, cardiac muscle, what it leads to is your heart's ability to pump blood because that's the function of the heart. So when we look at cardiac muscle and what it's doing, it's contracting, it's effectively getting the heart to pump, which then pumps blood throughout the body, which is which is a vital function of the cardiovascular system. All right, so that's cardiac muscle. Visceral muscle, or better known as smooth muscle, is named because it lacks striations. So smooth refers to its appearance. There are no striations. There's none of that banded pattern here, okay? It's a smooth spindle-shaped fiber, all right? Each one of these cells is going to be a spindle-shaped fiber. This term visceral, anytime you see the term visceral, think of like a gut, all right? Like guts. So, you get a visceral reaction to something, like you have a gut feeling towards something. So visceral really refers to, the inner lining of certain organs. But just keep that in mind that, smooth muscle, we're going to find this lining the walls of our hollow organs. Hollow organs would be, again, like your stomach or your urinary bladder, right? So It's something to keep in mind, right? Smooth muscle, we find it lining the walls of our hollow organs. GI tract would be a good example. Now, various functions of our smooth muscle, depending on the organ that we find the smooth muscle in, right? If you find the smooth muscle in your urinary bladder, well, that's necessary for the process of urination, right? When your urinary bladder, it actually expels the urine, the muscle there is contracting. Or let's take a look at the esophagus, for instance, right? The esophagus is conveying a food bolus down towards your stomach. And the process of those contractions that are taking place along your esophagus, they're known as peristaltic contractions. Okay, so the functions vary depending on the organ that we're looking at, but smooth muscle or any muscle at the end of the day, it contracts. It carries out the functions that it carries out by contracting. Does that make sense, guys? Are we okay with that? All right. I've got some quiet folks here, but hopefully everybody's following along. Definitely make use of lectures. I try to really emphasize things that are important. I say things that are, going to that you can be tested on over and over again. So if you hear me saying something over and over again, just perk your ears. All right, I'll try to allude to that information as much as I can to make sure you guys are prepared for meeting these outcomes. Okay, so back to the appearance part here. So one nucleus, no striations. Mononucleated, no striations, hence smooth. And then we look at its control and how smooth muscles are controlled. Also involuntary. We don't think about our stomach contracting. We don't think about our esophagus carrying out peristaltic contractions. So something to keep in mind, okay, under involuntary control. The only muscle here that's under voluntary control is going to be skeletal muscle. In fact, that's going to be the subject of what we talk about for the most part today. And we talk about cardiac muscle and smooth muscle more in detail when we get into the specific organs, as well as the heart when we talk about cardiac muscle. But today, and in our discussion on the muscular system, we focus mostly on skeletal muscle tissue, okay? All right, guys. So again, a lot to say on this slide, but hopefully you guys are okay with this. Any questions on this? No? We're okay? All right. All right, so moving right along. Now looking at the functions of muscular tissue. Some of these we've already talked about, but others we're going to actually look at right now. And bear with me one second. Where am I? We're going to get a more descriptive understanding of these functions here, okay? These functions of muscular tissue. Now, producing body movements, obviously we associate this with our skeletal muscles. Our bones are connected to skeletal muscles, which when they contract, they will pull on the respective bones. As far as it's a question in the chat box, are there gap junctions in smooth muscle? Well, it will depend on the organ we're looking at, but gap junctions, very generally speaking, they're basically just connections between neighboring cells that allow for communication of nutrients or movement of nutrients. It is absolutely vital that these exist in our cardiac muscle. Depending on the organ that we're looking at in which we find smooth muscle, they may or may not be present, but in cardiac muscle fibers, absolutely present, okay? So hopefully that answered the question. Susan. So other examples of smooth muscle, well, we have some examples like your like blood vessels, right? The walls of your blood vessels, or the walls of your stomach, or the your esophagus, like I was mentioning, your trachea, your windpipe, uterus, right? Smooth muscle contractions in your uterus, right? So these hollow organs are the ones that have a space within them. They're hollow organs, and the space could be fluid filled, it could be empty, but because of that space and the ability to uh, manipulate the volume of that organ or the space found within that organ, well, contractions can occur, right? Like your uterus, right? The uterine contractions, for instance, well, that's going to cause the uterus and the volume that is associated with it to decrease. So plenty of organs that have smooth muscle. Like I said, we'll look at as we talk about different organ systems, we'll refer back to, the function of smooth muscle in those particular organs. But generally speaking, like I said, all muscle cells that contract, okay? Alfredo, does that answer your question? I know there's a lot more to be said on that, but I hope I don't leave you hanging. All right, okay, good. So moving right along, So functions of muscular tissue: producing body movements. We know this. This works in conjunction with bones and joints. You guys covered joints at the end of last session. You guys talked about various types of joints like synovial joints, right? Synovial joints are going to be the structural designation of a joint. Functionally speaking, are synovial joints freely movable, kind of movable, or just immovable? Really movable, right? The freely movable ones. They're the ones that are going to like your elbow joint or your shoulder joint. Anytime you have these joints that are that allow free motion around them, it may be in a limited plane, but they are freely movable. They're synovial joints. So when we're talking about muscles, these muscles basically pull across these joints so that we can get our body to move. Right? The bones actually move as a result of the contraction of these skeletal muscles. Now, stabilizing body positions just kind of goes hand in hand with the first point, but your muscles, skeletal muscles specifically, they tend to remain in a contracted state at times, so that we can maintain body posture. Think about, you sitting up in your chair right now. You're not thinking voluntarily about your, back muscles to contract, but you're staying upright, right? Well, those muscles are in a state of contraction, not fully contracted, but a isotonic or isometric contraction that they're basically holding your body upright, right? And stabilizing your body position, okay? Storing and mobilizing substances within the body. Now, this is quite an interesting um, function here, because when we look at muscle tissue, well, some of these, some of the different types of muscle tissue, like cardiac muscle, this is pretty obvious, right? Cardiac muscle, when it contracts, the heart pumps, and when the heart pumps are able to mobilize a substance, that substance being blood. So that's pretty easy to infer there. But when it comes to storing, your muscle tissue, certain muscle fibers, like skeletal muscle fibers, they're actually able to store excess glucose. So when we ingest a meal, right, any meal you ingest, the glucose in there is going to be used up by your body for energy, but then you'll have extra that is left over. And if there's excess glucose left over, we want to store that, right? Because that's energy. We store it away as what's known as glycogen, and glycogen gets stored in your skeletal muscle tissues and in your liver. So something to keep in mind there, and we'll look at that again in a little bit more detail when we look at individual muscle fibers. But it's important to note that this is a function of muscular tissue. Mobilizing, again, a good example of that would be the heart. When it pumps, it mobilizes blood. When your blood vessels, when they constrict or dilate, they regulate the mobilization of blood. So good example there. Okay, everybody okay with that? With this third point, it's one that folks tend to miss out on, but hopefully that made sense. Everybody clear on that? Okay, so last one, generating heat. Generating heat. When muscles contract, they produce heat. This is why when you shiver, you warm up, right? Shivering is a involuntary act, but it's coordinated by your voluntary muscles, which are your skeletal muscles. And this is a part of, just regulating body temperature, a homeostatic measure, if you will. And the technical term for, your muscles generating heat when they contract, it's thermogenesis, okay? So why does this happen? Well, it helps to maintain our body's temperature, right? Our muscles start to contract, much more rapidly if you're getting cold, to warm up your muscles, right? Um, or warm up your body rather. So these are the functions of muscular tissue. They're kind of broad, and obviously, as we look at specific examples of certain things, we can get into one of these functions in much more detail. But be familiar with these functions, okay, guys? Everybody okay with these? These four that are listed here. All right, excellent.
Yes.
Okay, so what we're going to do, let's see, let me get to a couple more points here. We're going to take a quick break, okay? I try to give a break every hour, so a 10-minute break every hour, so that way, we can digest the information, go get some coffee if you need. I definitely will be making use of that break, so I need to grab some coffee. All right, let's just wrap up these next couple slides, and then we'll take a break, okay? So properties of muscle tissue. when you say properties of muscle tissue, well, these are shared properties. These are characteristics of muscle tissue, whether you're cardiac, whether you're smooth or skeletal. These are four properties that I need you guys to keep in mind. Electrical excitability. This is something I alluded to earlier on. Right? Electrical excitability simply means that your muscle fibers are able to respond to an electrical stimulus. Now, remember that shock that I was talking about, where along your neurons, you have this signal that gets sent along down a neuron, and then at the end of that neuron, it communicates it to a muscle. Well, it basically shocks that muscle. If your muscle fibers were not electrically excitable, that shock would do nothing, right? So because that action potential, technically speaking, that action potential, it is able to be received by our muscle fibers, we consider our muscle fibers to be electrically excitable. We can excite them by way of an electrical signal. The only two types of cells in the human body that are electrically excitable are going to be your muscle cells, your muscle fibers, and your neurons, okay? No other cell, right? So you can't have like a red blood cell that is going to respond to a electrical signal. It won't happen. The only types of cells in the human body that are electrically excitable, muscle cells and neurons, okay? And it makes sense because they have a very intimate relationship with one another. Now, contractility. This is another property of muscular tissue. In fact, this is probably the most important property of muscular tissue in terms of function. Your muscle tissue is able to, those cells that make up our muscle tissue, they're able to contract, they're able to shorten. And as a result of them shortening, they are able to pull on whatever they're attached to, right? If it's skeletal muscle fiber, well, it's going to be attached to bones, and it pulls on those bones, gets those bones to move, right? Now, contractility, so again, this refers to shortening. It only occurs in response to stimuli. So what is the stimulus that gets my muscle to contract? What would be the trigger, if you will, that will get my muscle to contract? The signal, if you will, the electrical signal, correct? Everybody on par with that? In order to get my muscle to actually contract, well, I need to send an electrical signal to it. And in response, that muscle will contract. So kind of goes hand in hand here. Does that make sense, guys? All right, excellent. Now, extensibility. Extensibility refers to the ability of your muscle fibers to actually stretch within limits, of course. So you can actually train your muscles to stretch to their outer limits, and you can increase those outer limits. But extensibility is simply the ability to stretch. Now, elasticity is the ability to return back to its original length and shape after having stretched beyond it or to its limit, or after having contracted. Well, much like a spring would, right? If you stretch out a spring, or like a rubber band. Actually, a spring is probably a better example. If you stretch out a spring, it's going to go back to its original position. Or if you compress a spring, it's still going to go back to its original position. So that's what elasticity refers to, okay? Is everybody okay with these four properties of muscular tissue? Are we okay with this? I want you guys to keep this in mind, all right? Keep these four properties in mind. Moving right along, some terminology. Terminology is huge in discussing the muscular system. Definitely be familiar with muscle fibers and that they relate to muscle cells. These cells, they come together to give you different muscle tissue. When we're talking about cardiac muscle tissue specifically, instead of calling them muscle fibers, we call them myocytes. Just some, semantic differences here. You don't have to, this is not like really a testable thing, but just be familiar with what muscle fibers are relating to. Also something that might help you out later on in, you're applying medical terminology, myo and sarco. Anytime you see these terms, what we're referring to is muscle. So myo means muscle, sarco refers to flesh, which indicates that it's talking about muscle. So if you see these terms, know that we're talking about muscles, okay? So what we're going to do now is hone in on skeletal muscle tissue and how skeletal muscle tissue actually contracts and carries out its functions at the cellular level. First, we're going to look at the cellular level because at the gross level, with the unaided eye, we can actually we know what skeletal muscles do. They contract and they pull on the bones to get them to move. In the case of, like, let's say your face, the skeletal muscles are actually attached to your skin. So rather than pulling on bones, they pull on your skin, and then you can make various facial expressions. But the idea here is that skeletal muscles, they contract to pull on things to get motion going, okay? How this happens, again, this happens in response to a stimulus, that stimulus being an electrical signal. So that's what we're going to look into as we go through these next couple of slides. First, what we'll do, though, is establish an understanding of the structural makeup of skeletal muscle tissue and how these cells are actually arranged within your skeletal muscles before getting into the functional components of it, okay? So how skeletal muscles are actually formed? Well, during embryonic development, what happens is these myoblasts, these precursor cells, they basically start fusing together. These myoblasts are basically stem cells that are, bringing to the equation their own nucleus, hence the multinucleated feature of our muscle fibers or skeletal muscle fibers. Remember, we saw that earlier on with the appearance in that table. The appearance of our skeletal muscle fibers is that it's striated, but it's also multinucleated. So the reason why it's multinucleated, well, because the precursor cells, these myoblasts during embryonic development, they basically brought with them their own nucleus and fused it together to give you this immature skeletal muscle fiber. Now, muscle fibers, in order for our muscles to get big, let's hone in on, let's just skeletal muscles. Your biceps are getting bigger. Say you've been going to the gym for a couple of weeks and you notice your biceps getting bigger because you've been doing bicep curls like crazy, right? And if that's the case, what's actually happening at the cellular level? Now, we can either have these cells growing in number, which would be considered hyperplasia, or we can have what's considered growing in size, hypertrophy. Which of these do you think is occurring here when your skeletal muscles grow? Are you getting more skeletal muscle fibers, or are you just getting them to grow larger? Grow larger, which is hypertrophy. So it's very important to keep in mind. If like, let's say you meet like a bodybuilder, like a meathead, and he comes up and he's like, yeah, I got a lot more. I'm working out for a period of time, and I have a lot more skeletal muscle fibers than somebody else, right? Or than I did before. He wouldn't. He'd be wrong there, right? You'd be wrong. You don't increase the number of muscle fibers. You simply get the muscle fibers to grow in size, and that concept is known as hypertrophy. Okay, hypertrophy. And the reason for this is our skeletal muscle fibers beyond or following embryonic development, they are no longer mitotic, right? So when you say something is mitotic, you guys remember mitosis from AMP1? When cells undergo mitosis, what happens? You have one
They
cell.
multiply.
Yeah, go ahead. Sorry, Jen, I didn't mean to cut you off. Go ahead.
Like I said, they multiply.
They multiply a number, right? So if something is amitotic or non-mitotic, right, it can't multiply a number. That's exactly what these skeletal muscle fibers are. They're amitotic or non-mitotic. They don't divide. So if we do want to get, our muscles to grow, we have to get the ones that already exist to grow in size, and that is known as hypertrophy, okay? Keep that in mind, guys. Hypertrophy. Skeletal muscles undergo hypertrophy, right? Cardiac muscles also undergo hypertrophy, and smooth muscle, there are instances where you do have mitosis occurring for smooth muscles, but for the most part, your muscle fibers are going to undergo hypertrophy as opposed to hyperplasia. Hyperplasia is when your cells start to increase in number. That's not what's happening here. Increasing in size instead, hypertrophy, okay? How do steroids make the muscles larger? Well, that's an interesting they basically increase the rate of metabolism for your muscles, which has to do with protein synthesis and all of that. That's something that hang on to that question, because we will see a connection with steroids and hormones when we get to the endocrine system towards the end of this session, when we talk about the endocrine system. So it's very, all of this is obviously part of the human body, so it's all going to be connected. But some of these very I guess specific connections we can definitely bring to the forefront when we get to the endocrine system and talk about something like steroids and how those kind of hormones can determine the growth of these muscles, but it has more to do with the activity that's taking place in these individual cells and the metabolism that's taking place in these cells to get them to grow larger. And once we get into the metabolism, it'll hopefully make more sense. So hang on to that question and we'll come back to that, okay? All right, so when we look at individual muscle fibers then, and then take a look at a cell, or I'm sorry, a muscle in its entirety, a skeletal muscle. What we see is that there's some connective tissue sheaths that wrap around our muscles. A mycelium is basically that connective tissue sheath. So when we see epi, anytime you see epi, think of something that's on the outside of something, okay? Peri is just in between, and then endo is going to be the innermost, right? And I'm going to show you guys a visual of this, but just picture like a cross section of, again, I pick on the bicep muscle because I think the images really just align well with that. But if you just took somebody's bicep muscle out of their arm, what you'd find is that surrounding that entire muscle is this connective tissue sheath that we call the epimysium. It's dense, irregular connective tissue, and it surrounds that whole muscle, right? And then muscles are arranged, our skeletal muscles are arranged much like tubes within tubes within tubes, right? Like this, right? Here's your massive tube, which would be your whole muscle. And I think we are looking at the bicep muscle here, your whole muscle. And then within that, you have these tubes, right? This tube that's found within our whole muscle is called a fascicle. But a fascicle is itself a bundle of tubes as well. These are the muscle fibers. So the arrangement is such that we have the whole muscle, which is made up of a bunch of these fascicles, and these fascicles are made up of a bunch of muscle fibers. Does that make sense, guys? Are we okay with what we're looking at here? I know it's jumping from the previous slide, going from just these words straight to an image. Hopefully I didn't lose anybody, but this I think does a better job of explaining just how the muscle is segmented. The whole muscle surrounded by that connective tissue sheath known as the epimysium. Found within the whole muscle are a bunch of these fascicles, right? A fascicle is, if I took one of these, I don't want to call them tubes here because they don't really look like tubes, but yeah, for sake of argument, we can just call them tubes, right? You took one of these out, that'd be a fascicle, but a fascicle is itself made up of a bunch of muscle fibers. A muscle fiber is the fundamental component. This is the cell. This is the muscle cell. Notice how it runs along the length of this entire muscle. This is where the action's happening in these muscle fibers. These are the key players here, okay? But each of these components, they're separated from one another by way of these connective tissue sheaths. So what surrounds the entire muscle is called the epimysium. What surrounds the individual fascicles is called the perimysium. And what surrounds the individual muscle fiber is called the endomysium. Okay? Everybody okay with that? All right. Now, you guys might, like, if you ever prepare like chicken or something like that, you know how you have to peel off that like layer of like, it's not really skin, but it's just like kind of like like saran wrap around uh, chicken, right? If you ever prepare chicken, sorry for any vegetarians or vegans in here. I'm sorry if this is unnerving for you. But if you've ever prepared meat, you peel that off. That's the epimysium, right? It's like it's kind of tough. Yeah, so yeah, and all three of these are wrappings of connective tissue. So what we can start to see is actually, the interplay between various tissue types here. Even though we're looking mainly at muscular tissue, there's obviously, of course, going to be some connective tissue involved. We also have some and blood vessels that are providing, nutrients to these muscle fibers. There's also going to be some nerves we don't see here, but yeah, that's the idea here, right? That the connective tissue sheets, they basically surround the components of our muscle fiber, or of our muscle, okay, our skeletal muscle. So here's a slide that basically, defines this. Hopefully the image helped make sense of this, and I do want you guys to be familiar with these connective tissue sheets. All right, the outermost, anytime you see epi, think of the outermost. Peri, in this case, is going to be the middle one. Endo, obviously, as the name implies, is going to be the internal one, the one that's closest to the individual muscle fiber, okay? And here's another image of exactly that concept. So what we can start to see is basically, just tubes within tubes, right? They kind of look like Twizzlers to me. But another key point to keep in mind here, and something we can actually see that we didn't see in the previous slide, is the presence of a motor neuron here, what's known as a somatic motor neuron. Now, why would we have a neuron here associated with this muscle fiber? What's that neuron doing? And why is it hanging around this muscle fiber?
It sends signals.
It's bringing a signal. Excellent. Electrical signal. It's shocking that muscle. What happens when this muscle gets shocked by way of this signal? How do muscles respond to electrical signals? What do they do?
Contract.
They contract. Excellent. So now what's going to happen is, if this individual muscle fiber contracts, it transmits that force to up that chain of structural arrangement. So muscle fibers contract, then the fascicle contracts, and then as those fascicles are contracting, then the whole muscle is contracting. Obviously, it doesn't take place, one after another. It takes place in seamless harmony, I guess you could say. It's not like a jittery contraction, but it's happening it's occurring initially at the muscle fiber level and being transmitted up to the whole muscle. So something to keep in mind there. And I think it's a good point right now to actually show you guys something I had pulled up, like complete anatomy. I use this software. I do have to pause the recording though while I show the. It is recording again now. So if you look at an individual muscle fiber, what we'll see is even within that muscle fiber, we find even more of these tubes. These tubes, they're basically protein filaments. They're known as myofibrils. And we'll look at what happens at this filament level, but this filament level is made up of these sarcomeres, right? And if a muscle cell is to contract, well, it's because of the activity of these sarcomeres that this muscle fiber is actually contracting, right? So that's what this next slide really indicates here. Here's your muscle fiber or fascicle, and a fascicle has a bunch of these muscle fibers within it. If we zoom into that, what we can start to see is the individual myofibrils within a muscle fiber. And you see a lot of activity, a lot of weird looking structures here, but at the end of the day, this is still just a cell. It doesn't look anything like that general visual that we saw of a cell in AMP1. You guys remember that generic depiction of a cell where you just had like, nucleus and you just had like a round pod-like structure. You had a bunch of organelles within it. This looks quite different, but it is still a cell and it is still going to have some of those components. Every cell is surrounded by a plasma membrane. Everybody remember that? Everybody okay with that?
Mm-hmm.
So in the muscle cell, however, we don't call it a plasma membrane. We call it the sarcolemma. It is still the plasma membrane, but we call it the sarcolemma. There's that prefix again, sarco referring to muscle. So the sarcolemma is just the plasma membrane that surrounds your muscle fibers. Now, in order for this muscle fiber to actually contract, there needs to be some activity that takes place at the sarcolemma, right? That activity is the electrical signal that gets sent from the neuron. It basically makes contact with the sarcolemma, and then as a result of that contact, the muscle fiber will start to contract. So it's very important that the barrier that surrounds our muscle, it plays a key role in getting that muscle fiber to actually contract. Okay. So is everybody okay with the sarcolemma and what it represents for our muscle fiber? We'll look at what happened, the activity here, but keep in mind that the sarcolemma is simply just the plasma membrane that surrounds your muscle fiber. And then when we look at inside the muscle fiber, what we see is that the sarcolemma, in certain instances, it actually invaginates into the muscle fiber itself. It actually penetrates in. Well, if you think about an electrical signal, like a shock coming down from a neuron, making its contact with the sarcolemma, and then going down into the fiber itself, then at that point, we can determine that this muscle fiber is being excited. So these little invaginations that we see here every so often, right, these are known as transverse tubules. Okay, we'll look at these in a separate image in a second, but I want you guys to keep in mind that the sarcolemma, it basically continues and invaginates into the cell. If it's carrying the electrical signal, well, that signal can then invaginate into the cell and basically travel into the cell and cause the components found within that cell to actually kick off that contraction process. And that's where these thick and thin filaments come into play here, okay? So everybody with me on that? I know that, visually placing yourself may be a little hard here, but is everybody okay with what we're looking at here?
Yes. Yeah.
Okay, so here's a better simplistic, as the caption indicates here, simplistic representation of the components of a muscle fiber. It looks quite different than what we saw here, but I think this image helps to illustrate what's going to happen or what is happening inside a muscle fiber when an electrical signal makes its way to that muscle fiber. Okay, so again, the outer border of our muscle fiber is a sarcolemma. Every so often, it penetrates into the cell, right? And that penetration into the cell is known as a transverse tubule. When we find the transverse tubule, on either side of the transverse tubule, inside the cell, we find these packets of calcium ions. So inside your muscle fiber, you find these packets. They're known as sarcoplasmic reticulum. Right? They're filled in with calcium ions. These calcium ions play a vital role in getting the contraction process kicked off, but we'll look at that in a second. I just want to identify structures right now, okay? So sarcolemma, plasma membrane, transverse tubule, and sarcoplasmic reticulum, filled in with calcium ions, all right? Other structures that we can find within our muscle fiber, we find these myofibrils, these long-stranded proteins that are made up of thick and thin filaments that are alternating. This is what gives the banded appearance, that striated appearance to our muscle fiber, are these alternating protein filaments that are running along the length of our muscle fiber. Does that make sense, guys?
Can you say it again?
So in our muscle fiber, we have these myofibrils. A myofibril is just basically long strand of proteins. But the way that these proteins are arranged, they're arranged in thick and thin filaments and alternating. So you have thick filament, then you have thin filament, thick, thin, and it just continues on. That's what gives the banded appearance. The thick filaments are going to be the ones that give the darker complexion. The thin filaments are going to give the lighter complexion. All right? Does that make sense, guys?
Mm-hmm.
Okay, so what we can then do is break apart this myofibril into regions that run from a particular part to another particular part known as a Z disc to Z disc. This is the sarcomere, okay? Now, if you think about, what's going to happen to our muscle fiber, it's contracting, right? That contraction is kicked off here at the sarcomere level, right? So whatever that signal that was coming from the neuron to the sarcolemma, it made its way into the transverse tubule. It basically made its way into the cell, right? And that cell is now excited to contract, but that contraction process kicks off here, these sarcomeres. They basically shorten, and as they shorten, the whole muscle fiber shortens. As the whole muscle fiber shortens, the fascicles shorten. As the fascicle shortens, the whole muscle shortens, right? But it's the sarcomere levels that we're really interested in to really get a granular understanding of muscle contraction. We okay so far? Questions, folks? Got a couple quiet folks here. I see some common names, and I appreciate that. And I also see some, I don't see some names, and that's not to say that everybody has to, jump into the chat box at every given moment. But I hope for those folks that aren't communicating, you are getting this. I don't want to leave anybody hanging. Okay. All right. So we'll look in the next couple of slides exactly the process, but the structures hopefully are you guys are getting more familiar with them. Couple more structures that I want to emphasize here. You see a bunch of mitochondria. Now, mitochondria. What is the importance of mitochondria? This is like the subject of a lot of memes.
ATP, energy.
Yeah, the powerhouse of the cell, right? So ATP, excellent, right? It produces a whole lot of ATP. So if our muscle cells are actually contracting, and they're contracting all the time, in order for them to do what they do, in order for any cell to do what it does, respectively, it needs energy, right? So muscle cells, they need a lot of energy. Energy comes in the form of ATP. ATP, for the most part, is made in mitochondria. So therefore, we have a lot of mitochondria in our muscle fibers. So we can see that here. Here's one mitochondria, here's another one. Two, I guess, is sufficient here. All right. And we also see myoglobin. Anybody, well, you guys may have heard of hemoglobin, right? So hemoglobin, anybody know the function of hemoglobin? What's
Yes,
hemoglobin?
carries.
Sorry, cut somebody off. Go ahead and say that one more time. Carries oxygen, right? It carries oxygen. Excellent. In order for mitochondria to actually produce the amount of ATP that it potentially can, it needs glucose, of course, but it also needs oxygen. So it makes sense that we have the muscle form of the hemoglobin present in our muscle fibers, known as myoglobin. Okay? So if I have a lot of myoglobin, right, that means I'm going to bring a lot of oxygen to this muscle fiber. If I can bring a lot of oxygen to this muscle fiber, that means the mitochondria can churn out a lot more ATP. And if I can churn out a lot more ATP, that means that this muscle fiber can contract a lot more. Does that make sense, guys?
I think I got lost a little bit. So hemoglobin carries oxygen, and that's important to the myoglobin. Why?
Okay, so sorry, let me jump back here. So hemoglobin, we know carries oxygen, right? Hemoglobin is found in our blood. Myoglobin is essentially like hemoglobin, except it doesn't carry oxygen throughout the blood. It's what carries and stores oxygen in our muscles. It basically does the same thing our hemoglobin does, but it does so in the muscle itself, right? It just has the oxygen that it can deliver to the necessary components within our muscle fiber. Does that make sense?
Mm-hmm.
Yeah, so if you have a lot of myoglobin, then we have a lot of oxygen storing capacity in our muscle, and therefore we can use that oxygen to produce a lot more ATP, and therefore that muscle can use that ATP to contract a lot more. And this is, yeah, there's a great question that came in the chat box. Is this why the muscles get tight during marathons, but eventually exhaust the myoglobin? It's one of the reasons. One of the reasons, yeah. We exhaust our ATP reserves. But this is also what's behind, certain muscles that have that are able to endure activity for longer periods of time. Whereas, like for instance, like your calf muscles, you can walk all day, and your calf muscles will be able to endure that, right? Whereas if you were to, like do bicep curls all day, that's probably not going to happen, right? You can't. Those muscles will exhaust a lot quicker. They don't have as much myoglobin in them, right? And this is also what lends itself to the understanding of fast twitch fibers and slow twitch fibers, which we'll talk about later on. But you guys may have seen, what's referred to as dark meat and white meat. That has to do with the presence of myoglobin. This is a pigmented protein. So darker meat is going to have more myoglobin, right? That's an indicator that it can use more oxygen, therefore produce more ATP, and therefore that muscle's going to have a lot more endurance associated with it. Does that make sense, guys? I know we're jumping a little bit here, but hopefully we're making some connections here. We good?
I feel connected.
Okay, excellent. That's good to hear. And the last component I want to mention here, glycogen granules. I mentioned this earlier on. Remember, we said that one of the functions of our muscle, or one of the characteristics of our muscles, I think it was on the function slide rather, storing and mobilizing substances, right? Glycogen is a storage form of something. What is glycogen a storage form of?
Damn.
Glucose.
Glucose, right? Glucose, excellent, right? So if I ingest a lot of glucose, some of that glucose is going to be used for the energy that I need, my immediate energy needs. And then the excess glucose is going to be stored away, right? Like I spent all day maybe looking for some food. I don't want to just waste that excess glucose. So my body stores it away. It packs it up as glycogen, basically links these glucose molecules together, packs it up as glycogen, stores it away in two places, in our skeletal muscles and in our liver. Excellent. So skeletal muscle and liver, two places where we store glycogen. Now, this is quite useful for our skeletal muscle because in order for mitochondria to actually churn out ATP, we need oxygen, but we also need glucose. So glucose is right here. It's just in the packed up form. We break it down into glucose and start to use that to catabolize and metabolize it rather, and then we can then churn out a bunch of ATP. So it kind of makes sense why we have glycogen granules in our skeletal muscle. They're the ones that need it a lot more. All right. Excuse me. All right, guys. So let's keep moving then. Hopefully this image, everybody's familiar with what all these tags that we're looking at here. We're going to now zoom into what a sarcomere looks like. So this part right here, the individual component of one of these myofibrils found within our muscle fiber, okay? A sarcomere basically just runs from Z disc to Z disc, and it's made up of thick and thin filaments, as well as some structural proteins. The thick and thin filaments, known as actin and myosin, or myosin being the thick filament, actin being the thin filament, these are the contractile proteins that are made that make up your sarcomere. Okay? Actin and myosin. Keep these in mind. Actin is a thin filament, myosin is a thick filament. Okay? And what we see, the arrangement of these thick and thin filaments in a sarcomere is you find a region where you see thin filament and no thick filament, right? And then you find a region of overlap. Right? Here's where they're overlapping. We have thick and thin filaments overlapping. And then you find another region that has just thick filaments. And on the other side, it just repeats. It's symmetrical here. So these are known as the zones that we can break our sarcomere into. You have the zone of overlap. You have the H zone. The H zone is simply where only thick filaments exist. And you have the I band. Excuse me. I band is where only thin filaments exist. Okay, so keep that in mind. There is a table on the next slide that states this, but visually, it helps to look at this visually and make sense of what's going to happen here when this sarcomere starts to contract, when it shortens, right? And it's because of the activity of these thick and thin filaments that this sarcomere will start to shorten. These thick filaments basically pull on the thin filaments, getting them closer to the M line. And what ends up happening is these thin filaments, they basically glide atop these thick filaments, getting closer and closer to the M line. So what do you think is happening to the H zone, the zone where there's only thick filaments? As this sarcomere is contracting, what's happening to the H zone? It gets smaller, right? To the point where it essentially disintegrates. The H zone, by definition, is where we only find thick filaments, right? But if these thick filaments are pulling on over them, like sort of like a blanket over them, if they're pulling these thin filaments over themselves, then what's happening is you no longer have a zone where there's only thick filaments. The thin filaments are basically gliding atop this region. As a result of that, the sarcomere is now shortening. Does that make sense, guys? Wish I had a visual to show this, but I think there's a slide that comes up that does represent this. But I want you to keep this concept in mind, though. The thick filaments and thin filaments, these are the ones that are responsible for the shortening of our sarcomere. And if these sarcomeres start to shorten all along that entire myofibril, then that whole muscle fiber is going to be shortening. If the whole muscle fiber is shortening, then the entire fascicle is shortening. It makes sense that other muscle fibers are also shortening at that point. And if the fascicle is shortening, then the whole muscle is also shortening. But it kicks off here at the sarcomere level. Does that make sense, guys? We're good with this? Okay, excellent. If there's any questions, feel free to ask. You unmute yourself at any point if you'd like. Some other proteins to make mention of here, we also see some structural proteins, major one being titin. Titin is this spring-like protein that basically maintains the structure of our sarcomere. We also see actually we don't see them listed here, but there are some other structural proteins that are coming up. They're listed here. I won't go into detail on these. I just want to mention that there are some structural proteins. We may talk about them as they come up in certain medical instances, but just be familiar with the fact that there are some structural proteins. It's the contractile proteins, however, that carry out the actual contraction of that sarcomere, myosin and actin. Which one of these is the thick filament and which one is the thin filament, guys? Myosin or actin?
Myosin is thick.
Okay, myosin is thick and then actin is thin. All right. Now you also have some other proteins here, troponin and tropomyosin. These guys, they're the regulatory proteins. So if I told you that, myosin basically pulls on actin to get it to glide atop itself, it needs to first come into contact with actin. But what we have in a muscle that's not contracting is these regulatory proteins acting. They're basically preventing that interaction from occurring. Hence the term regulatory, right? They regulate whether this interaction even occurs. So it's kind of like, myosin has a thing for actin, but troponin and tropomyosin, they're basically, in the way, right? They're blocking that. And what we need to do is basically get these regulatory proteins that convince them to get out of the way so that myosin can actually bind to the actin and get the actin to pull inward, right? And that's what we're going to look at in the entire process of contraction in a second. But is everybody familiar and okay with the function of contractile proteins, regulatory and structural proteins? Are we good with so far on this slide and what this is representing?
Mm.
We're going to talk more in detail about these individual proteins here and what actually happens in the contraction process, but just be familiar with the names at least, okay? A lot of terminology, be able to align them with the broad function before we get into the specific function. So troponin and tropomyosin, they're regulatory, okay? Now, here's a table that basically outlines a lot of what we just talked about in probably a lot fewer words, but take a look through that. I'm just going to jump ahead to the contraction process now, okay? So sorry if I'm jumping too fast. Yeah, here we go. Contraction and relaxation of skeletal muscle fibers. We said that myosin pulls on the actin and it causes those thin filaments to slide inward. As a result of this, the Z discs, those ends of that sarcomere, they get closer to one another, but effectively what we see is the sarcomere shortening. And because we have those structural proteins, we're able to transmit this force to the entire muscle. So as the sarcomere contracts, ultimately this results in the whole muscle contracting because of the presence of those structural proteins, like titin, like dystrophin, and like nebulin, what have you. But it's the sarcomere that's contracting initially. Okay. Here's an example of the sarcomere actually contracting. Right. Here we see a muscle that is somewhat contracted. We can see at the sarcomere level. We can see that there is the H zone highlighted here in green. But as this muscle starts to contract more and more, what you notice, and if you zoom in a little bit here, what you can notice is the thin filaments, they're basically moving atop the thick filaments more and more to the point where it's fully contracted, and now they're basically just hovering on top of the thick filaments entirely. That H zone disappeared. That area where there was only thick filaments doesn't exist anymore in a fully contracted muscle, right? So this illustrates the idea of the sarcomere being in a fully contracted state, which results in your muscle being in a fully contracted state. Does that make sense, guys?
Mm-hmm.
All right, excellent. Keep that in mind, okay? The H zone is what disappears, all right? Now, what actually happens such that we can get the H zone to disappear or the movement of these actin filaments inward? What actually takes place between the myosin and the actin? Well, this is what this contraction cycle outlines, right? We need ATP. That's something that we need in order for the activity of this myosin and actin to take place. We also need calcium, okay? Now, I mentioned calcium earlier on. Where was calcium in our muscle fiber?
In the sac.
In those sacs, right? Those sarcoplasmic reticuli. You guys remember
Mm-hmm.
that? On the previous slide, jump back if you have your slides pulled up. They're basically hanging out right outside of the transverse tubule. So you guys remember the transverse tubule was the inward penetration of the sarcolemma, right? I'll pull the slide up so to use your imagination here. But you guys remember this? The sarcolemma is just it continues and penetrates into the cell. And on either side of the cell, inside the cell, you find, either side of the transverse tubule, inside the cell, you find these sacs of sarcoplasmic reticulum, which are loaded with calcium. Okay? This calcium is important because in the contraction cycle, the calcium is what actually tells the troponin to tell tropomyosin to get out of the way so that this guy can bind to the actin. All right? It's really quite interesting when you consider this. Let's just jump back, show you what that looks like here. Okay? So this is a actin filament. And what you'll notice is there's a long stranded protein known as tropomyosin. Tropomyosin, regulatory protein, it's covering up those little black dots that you see there. Those are the binding sites. That's where myosin wants to make contact with actin. Now that's where it interacts with it. But tropomyosin is in the way. It's like a guard in the way, right? Tropomyosin only listens to troponin. So in order for tropomyosin to get out of the way so that myosin can bind, troponin has to tell it to get out of the way. What tells troponin to tell tropomyosin to get out of the way is the calcium. All right? So the calcium came from the sarcoplasmic reticulum. It came down into the cell and it basically bathed the inside of that cell and made contact with the troponin, these three proteins right here, those three dots really. It made contact with the troponin. Troponin then told tropomyosin to get out of the way. And now those binding sites are exposed. Now myosin can bind. If it binds, the myosin head binds, it's going to use the ATP to pull on the actin to get the actin filaments to slide inward. Okay, now that it's sliding inward, well, what's happening to the muscle when these actin filaments are sliding inward? What's happening?
Contraction.
Contracting, right? So in order for contraction to occur, you need the calcium. So calcium is a huge ion that we need to, it's a great signaling molecule, right? So whenever, or ion rather. So when we see calcium present in certain processes, we'll see that present as a signaling molecule. It basically signaled to the troponin to tell tropomyosin to get out of the way. Now myosin can bind to the actin filament, pull inward. This pulling inward is known as a power stroke. It pulls it in, and in order for it to detach so that it can do it again, you need to get a new ATP bound to the myosin head. Okay? Now the new ATP is basically a charged battery. ATP is like a battery, right? ATP would be the charged version of that battery. ADP is when it's dead, right? So put in a new battery, that new battery allows the myosin head to detach and attach again to another site and keep on going with that power stroke process as long as that signal is there. Okay? That signal coming from calcium. As long as that calcium is telling troponin to tell tropomyosin to get out of the way, these sites are exposed, and as long as we have enough ATP, this is going to occur. Are we good with that, guys? I would look at this process, and hopefully it's not daunting or anything like that. It's like it's really just like a choreographed process, right? Like it's you just have to know what's happening, what needs to be in place in order for certain things to happen. Troponin needs to tell tropomyosin to get out of the way. What tells troponin? Calcium. Calcium comes into troponin and says, hey, convince your man to get out of the way so that we can bind the actin to bind the myosin to the actin. All right. All right. So that's the contraction process. That's how our muscles contract at the sarcomere level. All right. What we're next going to do is take a look at the excitation part of it. Right. So, of course, in order for our muscles to actually contract, they need to first be stimulated to contract, and that stimulus takes place in the form of an electrical signal. We call that the excitation process. So we see that a similar image here as what we saw earlier on. Again, we see the sarcolemma, we see the sarcoplasmic reticulum, the calcium ions. But in order for these calcium ions to actually be liberated from these sacs, there needs to be the electrical signal. So here's the electrical signal. It's coming down the sarcolemma, basically shocking the muscle as it travels along the sarcolemma, right? And as it shocks it, it basically signals to the sarcoplasmic reticuli to release this calcium. So in order for calcium to be released, need the action potential, need the signal, okay? That signal causes the calcium ions to be liberated. They then make their way to the sarcomere level. Who do they talk to? They talk to troponin. Calcium signals to troponin. What does troponin do? What does troponin do to, now what does troponin tell troponin?
Troponin tells tropomyosin to get out the way.
Yeah, excellent. And then when tropomyocin's out of the way,
Then the
myosin,
contraction can
yeah,
happen and thin filaments can slide into the thick one and the H zone disappears.
excellent. So, all right, it sounds like I'm beating this over the head. It looks like you guys got this. All right, sorry for I tend to do that at times. Just let me know, all right? If you guys got it. It's hard for me to read faces because there are no faces. So just yeah, let me know if I'm going too much into detail on something. So hopefully everybody got this concept. Calcium is necessary, ATP is necessary, and in order for this contraction process to occur, but also the electrical signal that's necessary. Okay, and that came from a neuron, a nerve cell. So sure, we see it here traveling, but where did it come from? Well, it came from a nerve cell. And we'll look at that in just a second as well. But let's cover some of these slides before we get to that. We see the length tension relationship. This basically says that the length of the sarcomere prior to muscle contraction determines how strongly that muscle is going to contract. And this has to do with, if you're, let's say you're trying to throw a punch, let's say boxing and you're trying to throw a punch. If you're starting off with your arm fully cocked back, as opposed to, just right in front of your opponent's um, point of contact, right? Or face, wherever you want to make contact. Um, if it's fully cocked back, that's where you're gonna generate a lot more force. So the idea here is that there's an optimal length for your sarcomere to generate force. If it's overstretched, it's not going to be able to generate as much force, or if it's understretched, it's not going to be able to generate enough force. All right. So there's an optimal length here. It's called the length tension relationship. So here is some information on the neuromuscular junction, right? And the events that take place at the neuromuscular junction, well, these are the events that are going to produce that muscle action potential. They're basically what are going to shock and excite that muscle. In order for the muscle action potential to actually take place, there needs to be some voltage gated channels. Basically, these channels that allow for calcium to go in and out of the neuron and communicate to the neuron that we're exciting a post-synaptic cell. Right now, I don't expect that to make a whole lot of sense just yet. That's more in terms of the nervous system. But what I do want you guys to know is that calcium is involved here because, as we said, calcium is a signaling molecule. It was a signaling molecule in the contraction process in the muscle, but it's also a signaling molecule in the neuron itself. That neuron that's bringing that signal to the muscle, it also utilizes calcium so in order to release what's known as a acetylcholine neurotransmitter. Okay, this is a huge neurotransmitter that I want you guys to be familiar with. If you break that term down, neurotransmitter, what it's doing is transmitting that neural signal, that signal from the neuron to the muscle fiber. So acetylcholine gets liberated, it binds to the muscle on the sarcolemma, and this causes the sarcolemma to depolarize, which results in the calcium being released from the sarcoplasmic reticulum. A lot of terms here that we delve into in the nervous system, but from this particular slide, and what I want you guys to know is that acetylcholine is the neurotransmitter that's necessary for us to be able to transmit that signal from the neuron to the muscle fiber, right? Depolarize, we can talk about this term in more detail, but for right now, when you depolarize a membrane, what you're doing to that membrane and that cell effectively is getting it more excited, okay? Depolarization is associated with getting a cell more excited. So you start to depolarize that muscle. As you get that muscle more excited, that calcium gets released from the sarcoplasmic reticulum. We've seen this. So we know now what's happening as a result of acetylcholine being present. ACH is present, it's the abbreviation for acetylcholine, which results in ultimately the calcium being released from the sarcoplasmic reticulum. We know what happens to calcium when it gets released from the sarcoplasmic reticulum. It makes its way down to troponin. tells tropomyosin to get out of the way, and contraction happens. So kind of wrap this together then, right? So we can see that, you know, the nerve impulse gets brought by the neuron, the motor neuron. It releases acetylcholine. Acetylcholine then leads to the depolarization of the muscle, which leads to calcium being released from sarcoplasmic reticulum. Okay. I don't want to get too much into these channels just yet because there's, like I said, there's a whole chapter on this, and we talk about what these channels are, what these specific channels are. But if you just see what, you know, if you just want to get a general understanding of what channel is, it's basically a tunnel that allows for ions to go from one cell to another, right? The gate is indicated by, you know, what opens it up. Is it going to be a voltage difference or is it going to be a chemical? That part we'll talk about later on. Right now, I just want you guys to know that there are tunnels that allow for the movement of these ions that basically communicate to the neighboring cell or the next cell that, hey, you need to get excited, and you need to get excited and do this next. So that's essentially what's taking place here. It's a wordy slide. I don't really like wordy slides, but I try to highlight some things here that hopefully can help out here. All right.
So what did you want us to take away mainly from this slide?
Acetylcholine, the presence of acetylcholine. Acetylcholine,
Okay.
right? You
And
need
phenol is what's gonna get everything else to, that's the jump start.
Yep, that's the jump start. So here, a visual probably helps this a little bit better. Here's your neuron, your somatic motor neuron. It's bringing the signal. The point of communication is known as synapse, right? The little cleft in between the neuron and the muscle fiber, which is better known as the motor end plate part of the muscle fiber. That's known as the synaptic cleft. But what we see here in this image, I think it's a good image here, you see the nerve impulse coming down. As it comes to the end of the neuron, the synaptic end bulb, calcium floods in, right? Calcium makes its way in, and this calcium, again, is a signaling molecule. It signals to this end bulb to release acetylcholine. Acetylcholine is a neurotransmitter that floods the synaptic cleft, right? This area in between the neuron and the muscle fiber. It floods this synaptic cleft, but it binds to these receptors that are found on the motor end plate of the muscle. So you basically receive that acetylcholine, and that, right, that reception of the acetylcholine, it starts to get the muscle fiber more excited. It depolarizes it. Depolarization is associated with the movement of sodium going into the cell, but we'll talk more in detail about that later on. Right now, I just want you guys to know that it's depolarizing, and again, depolarizing is getting something more excited. So acetylcholine leads to the muscle fiber getting more excited, right? And as the muscle fiber gets more and more excited, it gets your muscle action potential is produced, right? The shock of that muscle is produced, and then that shock travels down to the T-tubule, tells the sarcoplasmic reticulum to release the calcium, and so on and so forth, okay? But none of that would happen if acetylcholine wasn't here, right? If you didn't have acetylcholine being released and then being received by the motor end plate, none of this would get kicked off, right? In fact, this is a point of you can administer drugs to inhibit the contraction of certain muscles. Botox is a good example of this, right? So Botox is basically, it results in the paralysis of a muscle. And muscle paralysis is basically, you know, inability to contract that muscle. The way that Botox does that is by occupying the space on these receptor proteins where acetylcholine is supposed to bind. So think about this. Your neuron brings that signal causes acetylcholine to be released, but there's nowhere for acetylcholine to bind. Is the rest of this going to occur?
No.
No, right? So if acetylcholine is being released, but there's nowhere for it to bind, then you're not going to excite the muscle fiber. Therefore, that muscle fiber is not going to contract. So a couple of things we need to keep in mind. Acetylcholine needs to be released, but it also needs to bind to the receptor. If the receptor is blocked by something else, it's not going to be able to bind. Or let's say that these receptors are degraded, and let's just say that they don't exist on this muscle, right? That's a separate condition. If they're just not present, then it's not going to excite that cell, right? And other conditions would be you're just not releasing enough acetylcholine. If you're not releasing enough acetylcholine, there's no way for you to transmit that signal to the muscle fiber, no matter what shock is going down your neuron. And if it's a skeletal muscle, you could be thinking about exciting or contracting your skeletal muscle, and that sends a signal. But if there's no acetylcholine being released, then there's no way for us to excite the muscle. So it's a huge regulatory point, especially in medical intervention, right? Does that make sense, guys? How acetylcholine plays a role in getting your muscle to contract?
Yes.
Oh, I'm sorry. Somebody said, can you use a red dot? I'm sorry. Yeah, my mouse doesn't. You're right. Absolutely. My apologies. Hopefully that's clear now. Because my mouse I changed the color of the mouse, but it changed back. I don't know why. It still went away. There we go. Good image to know, guys. Good image to know, right? Be familiar with this image. Definitely know the role of acetylcholine, right? So here's another really good image to know. It kind of encapsulates everything. It tells you, what's going on with the nerve action potential, the signal, right? What's happening with the acetylcholine being released by the neuron at the synaptic end bulb. Here in the synaptic cleft, acetylcholine receptor basically picks that up, and if it picks it up, and it's going to lead to depolarization of that membrane, of that muscle fiber, which is akin to saying that it's going to shock that muscle. That shock travels down the sarcolemma into the T-tubule, which causes the calcium to be released from the sarcoplasmic reticulum, floods the cell, makes contact with what it needs to, and gets contraction to occur, right? We good with that? I would take a
Yeah.
look at this image. It's a good image. This illustration really helps make sense of a lot of things. All right. Next, and we're going to wrap up with this. All right. We're going to talk about a very key component in all of this occurring, and that's the energy that you need in order for muscles to contract. The energy comes in the form of what?
ATP.
ATP, right? ATP, adenosine triphosphate, right? That's what it is, adenosine triphosphate. Adenosine triphosphate is the charged battery, right? It's a charged battery. When it's a dead battery, it's adenosine diphosphate, okay? So it's the energy currency of the body. So in order for us to actually produce ATP, let's just say we have a bunch of ADP lying around, in order for us to have ATP, what we need to do is add a phosphate to the ADP. Now, this takes place in a couple of different ways. As it relates to muscle cells, the way that our muscles get the energy that they need in order to contract, well, there's three different ways, and it depends on the duration of that contraction. So, and the duration of the activity. The first of these is creatine phosphate. Next is the anaerobic glycolysis, and lastly, we see cellular respiration. Let's say you just drop down and started doing a push-up. For the first 15 seconds or so, your body's going to use creatine phosphate reserves to deliver the phosphate to the ADP to get it to become ATP for about 15 seconds or so. So let's say you keep doing those push-ups. Let's say you're about a minute and a half in, right, of doing push-ups. Beyond 15 seconds or so, you're now tapping into this process known as anaerobic glycolysis. Anaerobic, what does that tell you?
Without oxygen.
Without oxygen, right? So this process doesn't rely upon the presence of oxygen, but we're now using glycolysis, anaerobic glycolysis, which is breaking down glucose to give us the ATP that we need. Still not we're not getting a lot of ATP here, but more than we would with creatine phosphate. And that makes sense because the duration is increasing. And then lastly, let's say you keep on doing those push-ups and you're at it for like 10, 15 minutes. At that point, you're using cellular respiration. Now, this process does rely upon the presence of oxygen, right? So it depends on the extent of that activity. The longer that activity or the muscular contraction is taking place, you'll start to rely more upon these later mechanisms. Cellular respiration is what's associated with cardiovascular training, for instance, right? And that makes sense because when you're doing cardiovascular training, let's just say you're running a marathon, you're going to be breathing pretty heavily. You need oxygen, right? Your muscles need the oxygen in order to produce that ATP, but they're producing a lot more ATP, right? Whereas if you're just doing bench press or strength training, right? Let's just say you're doing a set of squats. Well, that likely won't take place for longer than 2 minutes, and you're using anaerobic glycolysis. So, for those that may be familiar with any kind of like exercise terminology, you may have heard of things like anaerobic training or aerobic training. Well, the difference is found in how your muscles use or produce the energy needed to get them to contract. All right. The next couple of slides really just go into detail on these three processes. So again, creatine phosphate, this provides energy for about 15 seconds, immediate source of energy. There's the creatine phosphate reserves in your body basically deliver phosphate groups to ADP, getting those ADP molecules charged into ATP. Now our muscles can use ATP. So this is an immediate form of ATP production. Not a whole lot of ATP though, right? So beyond this, let's say you continue beyond 15 seconds or so, well, at this point, you're now relying upon anaerobic glycolysis. And this is good for about 2 minutes. Anaerobic glycolysis leads to the production of lactic acid. Lactic acid manifests itself as soreness, right? So the more lactic acid you have accumulating in your body, the more it's going to start to your muscles are going to start to feel sore, right? So this is anaerobic glycolysis, and this is good for about 2 minutes. Beyond 2 minutes, now you're tapping into cellular respiration or aerobic respiration, right? And this relies upon oxygen, but this also produces a lot more ATP. This allows your muscles to be able to carry out their activity for several minutes to hours, depending on the muscle and depending on the activity as well. So just something to keep in mind here, okay? That cellular respiration gives you a lot more ATP than either of the other two, but it's necessary because this is going to align with longer duration of energy. All right, so are we okay with how our muscles get the ATP that they need in order to carry out the contraction that they carry out?
Yeah.
Sound like kind of an uncertain yeah. I'll take it though. It may just be a tired yeah.
I might not understand every single one. I understand probably the last two, and then creatine phosphate, I probably just have to read on my own, but I got the words down.
Okay, excellent. So yeah, creatine phosphate is basically just you have these vehicles that are carrying around phosphate groups, right? So phosphate groups get delivered to ADP. ADP is lacking a phosphate, and if you give that ADP and phosphate, it turns into ATP, adenosine triphosphate. Now it's charged. That's essentially what's happening here. Creatine phosphate just delivers the phosphate to our ADP molecules, getting them to ATP molecules. Now our muscles can use the ATP that's present. So let me ask you guys a quick question on this then. ATP specifically, where is it used in the contraction process?
In the contraction process.
The contraction process. So you guys remember the contraction. Contraction entails the binding of the myosin head to the actin. Then you have the power stroke, and then you have the detaching of the myosin head so that it can attach again and carry out another power stroke. Where in that process is ATP required?
It pulls back myosin.
Yeah, it detaches it so that it can do it again. That's exactly correct. So let me go and show you that really quickly. So jog your memory on this. Here's a myosin head that if it was charged, right, only then can it actually become in or get into a position that it can bind to the actin filament. And it's basically extracting the energy from ATP, using the energy from this battery to carry out this whole process. But in order for me to get it to detach, because right here you see it pulled in, but it's still attached. In order for me to get it to detach and do that process over again, I have to give it a new ATP. Right. So it's the detaching that ATP is absolutely necessary for. Right. Without detaching, you're not going to be able to reattach. And there's plenty of these myosin heads. It's not just one of them. There's plenty of them. So it leads yourself to the understanding that you need a lot of ATP to carry out continual contraction, and that is the case. So you do need a lot of ATP. Energy is very important. All right, so That's why we have, these different methods. Our body's adapted to use these different means to give us the energy that we need. All right. So this next slide here is relating to muscular fatigue. How do muscles tire out and cease contraction? Well, fatigue it can be muscular fatigue at the muscular level where the muscle itself just cannot do it anymore, or it can be central fatigue where your central nervous system is saying, hey, sure, you may be able to push yourself and contract your muscle a couple more reps or what have you, but you're going to cause damage. So that's central fatigue. Muscle fatigue is when the muscle itself, again, is unable to do so. And this can happen as a result of no more calcium from sarcoplasmic reticulum. There's no more creatine phosphate, oxygen, or nutrients necessary for metabolism to occur. Or if you have a lot of lactic acid building up and a lot of dead batteries accumulating, right? That tells your body that, hey, you've contracted your muscles a lot and you produce a lot of waste products. Cut it out at this point. Your muscles are basically going to say, okay, there's no energy left and there's a lot of waste products accumulating, right? And insufficient release of acetylcholine and neuromuscular junction. Well, we know why what happens when this happens, right? There's no excitation taking place then. If there's no acetylcholine, there's no way for me to excite the muscle fiber. Make sense, guys? How muscle fatigue comes about?
Mm-hmm. It's coming together.
Yeah, that's good. That's good to hear. Go through this a couple times. Maybe once you guys do the EDAPT concepts, it'll start to tie in together. So real quickly on central fatigue, then central fatigue, this is when the central nervous system, your brain basically tells, it's cutting out the exercise and the contraction of those muscles. It's basically saying, to prevent any further damage to your muscles, cut out the exercise. This is something that you can overcome. You can overcome this. This is why you see, like, on Instagram and stuff, you see people like grinding away at like heavy weights and stuff like that, and they're overcoming central fatigue. But once you reach the point of muscular fatigue, this is going to be difficult to overcome. You're not going to be able to overcome the muscle itself no longer being able to contract. You may be able to overcome your brain telling your muscle to stop contracting, but not the muscle itself ceasing to contract. All right. So, all right, and this last slide, and I'll wrap it up here, guys. We'll pick up on control of muscle tension tomorrow, but this last slide has to do with oxygen, right? Oxygen is used in various ways, but especially with cellular respiration, we do need this. But let's say you've been running for several minutes to hours, you're definitely relying upon cellular respiration, which requires oxygen. So after running for that long, you're likely breathing pretty heavily. Reason why you're breathing pretty heavily is because you're trying to replenish your oxygen reserves. You're trying to get oxygen back into your muscles, into your myoglobin, so that they're nourished and they're able to uh, uh, repeat the process if necessary. And then the extra oxygen, you can replenish creatine phosphate reserves, and you can also convert lactate into pyruvate, which is necessary in order for us to further metabolize those waste products. And again, reloading O2 onto myoglobin. Remember, myoglobin was the muscle version of hemoglobin, just stores and gives oxygen when it's needed into the muscle. So why oxygen consumption after exercise is necessary. Okay. All right, guys. So I will wrap it up here for today.